Welcome to rapid series of CET biology. Next topic is biomolecules. There are two types of molecules. One is micromolecules, second is macromolecules. What are macromolecules? Those are the larger molecules and micromolecules being the smaller molecules. Now we are going to see three important macromolecules. First are polysaccharides. What are polysaccharides? They are also called as carbohydrates. They are the polymers of monosaccharide. Polymers means what? There are many units, repeating units present of the monomers. The monomer of polysaccharide is monosaccharides. Next, polypeptides. They are also called as proteins. They are polymer of what? Amino acids. Third is polynucleotides, means DNA, RNA. The polynucleotides are also called as nucleic acids. These are the polymer of nucleotides. Now, the figure 6.1 which is given in your textbook, you have to prepare that the figure is of biomolecules in living system. Next is the carbohydrates. First, we are going to study about carbohydrates. What are carbohydrates? Already we have seen they are polymer of monosaccharides. Monosaccharides means simple sugars. Carbohydrates are hydrates of carbon. They are form of carbon, hydrogen and oxygen. Molecular formula of carbohydrate is CH2ON. Hydrogen and oxygen ratio is same like of water, that is 2:1. Here the table which is given 6.2 classification of carbohydrate with example. Mostly questions are asked from that table where they have given the classifications of monosaccharide, disaccharide, polysaccharide along with their example and their common names also. First we are going to study monosaccharides. What are monosaccharides? Monosaccharides means simple sugar, the monomers. They are crystalline, sweet, they are soluble in water, they can mix in water. Rare in nature, they are not found in nature. Mostly the disaccharides are found in nature. Classified as triose, that is 3 carbon, tetrose 4 carbon, pentose 5 carbon, hexose is 6 carbons. Next, they are reducing sugar. They are, all the monosaccharides are reducing sugars. If they contain aldehyde group, that is CHO group, they are called as aldoses. If they contain keto group, that is C double bond O group, then they are called as ketoses. The examples of aldoses are glucose, xylose, and example of ketoses are ribulose and fructose. We are going to see the examples of monosaccharide. First is glucose, second is galactose, third is fructose. Glucose is the most important fuel of living cell. The concentration of glucose is 90 mg per 100 ml of blood. Glucose is also called as the blood sugar. It is of 6 carbon. Next is galactose. Galactose is also of 6 carbon, that is hexose sugar. Galactose plus glucose gives lactose, which is the milk sugar. Third is fructose. It is also 6 carbon but it forms 5 carbon ring structure. Fructose is fruit sugar. Fructose plus glucose gives sucrose, which is also called as cane sugar. This is about the monosaccharides which cannot be hydrolyzed further, means it cannot be broken down further. Second is disaccharide. When two molecules of monosaccharide are jointed together by the condensation reaction, then it is called as, the structure formed is called as disaccharide. Bond present between two monosaccharides are called as glycosidic bonds. So in disaccharide, the bond which is present between two monosaccharide unit is called as the glycosidic bond. The reaction of formation, that is joining of two monosaccharide, is called as condensation reaction. Example, sucrose is reducing sugar, lactose maltose is not reducing sugar, which are examples of disaccharides. The disaccharides are soluble in water, that is they are mixable in water, but due to their large size they cannot pass through the cell membrane. So they need to be broken down into monosaccharide to take up in the cell. Broken down into small intestine during digestion. Here one glucose is jointed to another glucose molecule to form maltose. This bond here which is present is called as the glycosidic bond, which is present between first and sixth carbon, so it is called as 1,6 bond. Now what is alpha and what is beta? When the bond is formed below, then it is called as alpha glycosidic linkage. When the bond is formed like this on the upper side, then it is called as the beta glycosidic linkage. Here in maltose we get to see this bond downside, so it is called as alpha 1,6 glycosidic linkage. Next we are going to say is polysaccharide. Likewise, how in disaccharide one condensation reaction takes place, in polysaccharide series of condensation reactions takes place. The series of condensation reaction are called as polymerization. Polysaccharides are broken down into simple sugars by hydrolysis. First, starch. What is starch? Starch is stored in plants. It is storage material which is present in plants. made up of alpha glucose. Starch has two forms. One is the linear form, that is unbranched form. Another is the branched form. The linear form, which is unbranched, is called as amylose, and the second is called as amylopectin, which is the branched structure. Glycogen is amylopectin in nature, means it is always found in the branched form. Okay. Nextly, the short distance is present between branches. It is also like the starch amylopectin, but the distance between the branches is very short. Lastly, it is stored in animals' liver and in muscles. Both these are the polysaccharide of glucose. Lastly, cellulose. It is also polysaccharide of glucose, but of beta glucose. Straight. Next, it is found in the cell wall of plants. Next point is lipids. What are lipids? They are soluble in organic, that is non-polar solvent. The lipids hydrogen and oxygen ratio is greater than 2:1, that is greater than that of water. There are three types of lipid. One is simple lipid, second is the compound lipid, and third one is the derived lipid or sterols. Now we will see two types of lipids. First are saturated lipids, which have no double bond, example palmitic acid, stearic acid. Second is unsaturated lipid with one or more double bond, example oleic acid, linoleic acid. Now first we are going to study simple lipids. First example is fats, second example is wax. Esters of fatty acids with glycerol are called as fats. Example triglycerides, where we find 3 fatty acid chains along with 1 glycerol. If there are 2 fatty acid chain with 1 glycerol, they are called as diglycerides. If 1 fatty acid chain with 1 glycerol, then they are called as monoglycerides. Unsaturated fatty acids, they are usually liquid at room temperature, and so they are commonly called as oils. Next is high calorific value. These fats, the simple lipid first example fats, they have high calorific value. So in plants they are stored in seeds, and in animals they are used as storage material in adipocytes. Second is wax. They are esters of long chain of fatty acid with alcohol. See the difference here in fats, that is esters of fatty acid with glycerol. In wax, these are esters of fatty acid with alcohol. Found in blood, gonads and in sebaceous glands. They are solid at room temperature. We have seen that these are liquid at room temperature, these are solid at room temperature. Second one is compound lipids. Esters of fatty acid, if they are found with other molecules like phosphate, sugar, then they are called as compound lipids. If they are found with phosphate, then they are called as phospholipid. We know phospholipid is usually the component of cell membrane. Example lecithin. Second is, if the fatty acids are found with sugar, then they are called as glycolipids. These glycolipids are mostly found in brain, and so they are called as cerebrosides. Third one is the sterols. They are also called as derived lipids. For example, cholesterol belongs to this group. Fused hydrocarbon ring and a long hydrocarbon chain. Example, cholesterol, which is found in animal, particularly more amount of cholesterol is found in the nervous tissues. Cholesterol is not found in plants. In plants, phytosterols are present. For example, yam plant, which produces diosgenin, is used in preparation or in manufacturing of the birth control pills. Molecules of proteins. These are the nitrogenous substances. Usually, protein contain 100 to 3000 amino acids. The bond between two amino acids is called as the peptide bond. 20 amino acids are found in the protein. These are the two amino acids. First amino acid and second amino acid. The bond between these two is formed by elimination of water molecule. That is, CONH bond, which is formed here, is called as the peptide bond. Now we are going to see the structure of protein. First is the primary structure of protein, second is the secondary structure, third is the tertiary structure, and fourth is the quaternary structure of protein. What is primary structure of protein? The linear chain of amino acid 1, 2, 3, 4, n number of amino acids. When they are linked together by the peptide bond, the structure is called as primary structure of protein. The terminals where first amino acid is found is called as the N terminal, and the last amino acid which is found is called as C terminal, because we know that the first terminal where first group is NH2 group, that is amino group, so that is called as N-terminal, and the last amino acid which has 3 carboxylic groups, so it is called as the C-terminal. Secondary structure, the examples are alpha helix or beta pleated structures. These are the alpha helices, these are the beta pleated structure. Stability of these structures are due to the additional hydrogen bond which are present along with the peptide bond. Example, keratin in hair is the example of secondary structure. Helix is right-handed, then it is called as alpha helix, and if the helix is left-handed, then it is called as the beta helix. Third is the tertiary structure. When the amino acid chains or the peptide chains are bounded or are looped and twisted upon each other and give a 3-dimensional structure to the protein, then that structure is called as the tertiary structure of protein. Example, myoglobin, which is also called as muscle protein. Last is the quaternary structure of protein, where more than one polypeptide chain or one peptide chains are jointed together to each other. The protein which is found is called as the quaternary structure of the protein. Protein subunits are held together in this quaternary structure. Example, hemoglobin has 2 alpha chains and 2 beta chains. Proteins they are reactive, they are amphoteric in nature, means they can function as acid as well as base. They have an isoelectric point. What is isoelectric point? Where the net charge of overall protein is 0. Basic protein, if the number of amino acid, that is the basic amino acids number in the protein is more, the protein get characteristic positive charge, and that protein is called as the basic protein. Example, histones are the basic proteins. Acidic protein, if the number of amino acid, that is acidic amino acids in the proteins is more, the protein get overall negative charge, and that protein is called as acidic protein. Example, blood proteins are mostly acidic. Next point is classification of protein. There are 3 types of protein. One is simple, second is conjugate protein, and third is the derived protein. First, simple protein. Simple protein, when they are broken down, they will yield only amino acid. They are soluble in water. Example, histone protein, egg albumin, serum albumin, which are made up of only protein molecules, are called as simple proteins. Second, conjugates. What are conjugated protein? When they are broken down, they will yield amino acids along with the another non-proteinic group. Example, globin protein, that is hemoglobin. In hemoglobin, globin is the proteinic part and heme is the prosthetic group which contain iron. Mucoproteins. Second, mucoprotein, they are carbohydrate plus proteins, means on breakdown they will yield protein along with carbohydrate. Example, mucin of saliva, heparin of blood are examples of what? Mucoproteins, that is the conjugates. Third, lipoprotein, which are made up of lipids plus protein, means when they are broken down they will give protein part along with the lipid part. Example, these proteins are found in brain and milk. Third is derived protein, which are not found in nature. Example, metaprotein and peptones. Now the last group of macromolecules is nucleic acids. What are nucleic acids? The DNA and RNA, that is deoxyribonucleotides and ribonucleotides, or ribonucleic acid and deoxyribonucleic acids are called as the nucleic acids. First of all, we will see the nitrogen bases which are present. The nitrogen bases are classified into 2 groups, purines and pyrimidines. Purines are double ring, pyrimidines are single ring. Purines includes adenine and guanine, pyrimidines include cytosine, thymine, uracil. In DNA, cytosine and thymine are present, and in RNA, thymine is substituted by uracil. DNA, that is nucleotide, it is made up of polynucleotide. What is polynucleotide? Made up of many units of nucleotides. Single nucleotide includes sugar, phosphate and nitrogen bases. If there is no phosphate group, if there is only sugar and nitrogen base, then the structure is called nucleoside and not nucleotide. In nucleotide, sugar is present, phosphate is present and nitrogen base is present. The sugar which is present in DNA is deoxyribose sugar. In nucleoside, no phosphate group is present. What is Chargaff's rule? We know that adenine always pairs with thymine by 2 hydrogen bond and guanine always pairs with cytosine by 3 hydrogen bond. One purine always forms bond with another pyrimidine. So, the Chargaff's rule states that A plus T upon G plus C is equal to 1. We are going to see the structure of DNA. The structure of DNA was given by Watson and Crick. The structure of DNA was given by Watson and Crick. In Watson and Crick model of DNA, we will find that the two strands of DNA, they are always anti-parallel to each other and they are complementary. What is anti-parallel? If one strand runs from 3' to 5' direction, the another strand will run from 5' to 3' direction. This is called as anti-parallel. What is complementary? The complementary means A will always form 2 hydrogen bonds with T and G will always form 3 hydrogen bond with C. You will never see A binding with C or G binding with T. No, A will always bind with T and G will always bind with C. DNA model of Watson and Crick also suggests that one group is made up of 3.4 nanometer and this one group includes 10 base pairs, that is 10 nucleotide base pairs. Here the distance between two base pairs is 0.34 nanometer. Bonds between nucleotide is phosphodiester bond and bonds between nitrogen bases are called as the hydrogen bond. Acid is RNA. RNA is ribonucleic acid. Genetic material in most of the viruses is RNA. Sugar which is found in RNA is ribose sugar. Uracil is present instead of thymine. So, here always A will pair with U, that is uracil, and G will pair with C. There are three types of RNA, that is messenger RNA. There are three types of RNA. First is mRNA, which is also called as messenger RNA. Second is rRNA, which is also called as ribosomal RNA. And third is the tRNA, which is also called as transfer RNA. First, mRNA is the carrier of genetic information which carries information from DNA to RNA. It is linear, it occupies 3% of the volume of total RNA. The functional unit of mRNA is called as cistron and the synthesis always begins from 5' and ends at the 3' end. Second is rRNA, which is also called as ribosomal RNA, makes up 50 to 60% of ribosomes and 80 to 90% of the RNA synthesized in nucleus, and this was discovered by Corland. Third type of RNA is tRNA, which is also called as transfer RNA, which has important role in translation, that is protein synthesis process. They are small, 70 to 80 nucleotides, clover leaf structure. It looks like the clover leaf. 4 arms. First is the amino acid recognition arm, which is also called as DHU arm. Second is the amino acid binding site. Third is the anticodon loop, which binds with the codons which are present on mRNA during translation. And fourth is the ribosome recognition site. It is enzymes. What are enzymes? They are biocatalysts. What are biocatalysts? Which will increase the rate of chemical reaction. Without enzyme, the rate of chemical reaction will proceed in very slow rate or very slow speed. Enzyme act on a suitable substrate and convert that substrate to the product. There are two types of enzymes. Endoenzymes, which act within the cell in which they are synthesized. Exoenzymes, which act outside the cell from which they are synthesized. Types of enzymes. First, they are purely proteinaceous. Second type is the conjugate enzymes. What are conjugate enzymes? The enzymes which has prosthetic group along with the proteinic group are called as the conjugate enzymes. Conjugate enzymes, the prosthetic group may be coenzyme or cofactor. If the prosthetic group is organic and which is tightly bound to the proteinic part, then they are called as coenzymes. Example, NAD, FAD are coenzymes. Cofactors, the inorganic part which is loosely bound to the proteinic part are called as the cofactors. Example, copper, zinc, etc. are the cofactors. Iron is cofactor in enzyme catalase and manganese is a cofactor in peptidase. Enzymes are destroyed at high temperature, that is above 60-70 degrees Celsius, and they are inactive at the low temperature. What is the optimum temperature of the enzyme? The highest activity which the enzyme shows at that temperature, the temperature is called as the optimum temperature. The maximum efficiency of the enzyme is seen at the optimum temperature. Same, what is optimum pH? The pH at which the enzyme shows maximum activity or the efficiency rate, highest efficiency rate, that point is called as the optimum pH. For pepsin which is present in stomach, pH is 2, optimum pH is 2, and for trypsin which is present in duodenum, the optimum pH is 9.5. Now we will see classification. 6 types of enzymes are there or 6 groups of enzymes are there. Suffix ASE added to the group. First is oxidoreductases, which carry out oxidation reduction reactions, both the reaction or redox reactions. Example, alcohol dehydrogenase, Second, transferases, which transfer the group from one molecule to the another. Example, glucokinase. Next, hydrolases, which carry out hydrolysis reaction. Example, amylase, protease, which break down protein, which break down amylose. Next, fourth one is the lyases. What are lyases? Elimination reaction causing which can leave the double bond. Example, aldoses, dehydratase. Fifth is isomerases, means which are enantiomers of each other. They cause structural rearrangement. Example, epimerases or isomerases. Last group is ligases, which carry out joining of the molecules by forming covalent linkage. Example is pyruvate carboxylase. This is the model of enzyme activity. First model is lock and key model and second is induced fit model. Most accepted model is the induced fit model. First we will see what does lock and key model suggest. It was given by Emil Fischer in year 1894. It tells us that substrate fit in the active site of the enzyme. Like the substrate is compared with key, active site is compared with the whole of the log and enzyme is the log. The key fits actively in the whole of the log. That is, the substrate fits actively in the enzyme or the active site of the enzyme. Second, induced fit model. It was given by Koshland in year 1959. It suggests that substrate induces a conformational change in enzyme. This is the more accepted model. Here we can see this, the enzyme, the substrate, when they come in contact, when they come in contact, the enzyme changes its active site or makes the temporary change in the active site so that the substrate can be bounded more tightly with the enzyme to produce the product. And once the product is produced, the enzyme regains its original shape and the product is released free. Now, next point is the factors affecting enzyme activity. First factor is concentration of substrate. Rectangular hyperbola is obtained when we plot the graph of substrate concentration against velocity. Initially, the velocity increases along with the increase in the substrate concentration, but at point when the velocity reaches its maximum speed, substrate concentration doesn't matter. Whether we increase the substrate concentration, at Vmax there will not be any further increase in the velocity of the enzyme activity. The point where maximum velocity is seen is called as the Vmax. Half Vmax means the half of the maximum velocity, and the substrate concentration which is found at half Vmax is called as Km. Michaelis Menten constant. It is the substrate concentration to produce what? To produce half of the maximum velocity. Measures of enzyme substrate complex. What does Vmax or what does Km value gives? Km value gives the measure of enzyme substrate complex, that is the affinity between enzyme substrate complex. If there are low Km values, it will suggest that the enzyme substrate complex has high affinity for each other. If the Km values are high, it will suggest that there is weak affinity between the enzyme substrate complex. Second is enzyme concentration. Enzyme concentration is directly proportional to the concentration of substrate and it is directly proportional to the square root of concentration of enzymes. Third is temperature and fourth is pH. As we know, we have already seen, enzyme shows maximum activity at the optimum temperature and optimum pH. If we see about the temperature, enzymes denature at higher temperature and they become inactivate at the low temperature. Same for the pH, the highest rate is seen at the optimum pH. Point is metabolism. What is metabolism? It is sum of all the chemical reaction which takes place in cell. There are two types. One is catabolism, another is anabolism. Catabolism is the breakdown of complex molecule into simple forms. For example, starch to glucose is the catabolic reaction. Here energy is released, that is we gain energy. What is anabolism? It is biosynthesis of molecules. Synthesis of complex molecule from the simple substance is called as anabolism. For example, synthesis of protein from amino acid is called as anabolic reaction. Here we need to invest energy, that is the energy is consumed in anabolic reaction. So the sum total of anabolic and catabolic reaction are together called as metabolism. What is metabolic pool? It is the reservoir of biomolecules. The reservoir of biomolecules is called as the metabolic pool. Lastly, we are going to see secondary metabolites. These are the small organic molecules produced by bacteria, fungus and plants which are important for the human welfare. First, terpenes, they are made from mevalonic acid, they are also made up of carbon and hydrogen. Second is phenolics, which are made from Simple sugars along with benzene ring, which also contains hydrogen and oxygen. Last is the nitrogen containing compounds, which also contain sulfur in small amount. Example is morphine, which is the first alkaloid obtained from plant Papaver somniferum, which act as a pain reliever. So here we have completed our second topic, that is biomolecules from the CET series.